The search for alien life just changed. Not in the way people have been saying it would change for decades, not with a radio signal from a distant star, not with a photograph of something moving, not with a spacecraft returning samples from a Martian trench. It changed quietly, in a spreadsheet of spectral data from a telescope floating 1 million miles from Earth, in numbers that took months to interpret, in a molecule that smells like the sea. The molecule is called dimethyl sulfide. On Earth, it comes from living things, almost exclusively. Marine microbes produce it when they decompose. It drifts up from the ocean into the atmosphere and seeds clouds. It is one of the chemicals that makes a coastline smell like a coastline. And astronomers using the James Webb Space Telescope have found its spectral signature in the atmosphere of a planet 120 light-years away called K2-18b. The team, led by Niku Madhusudan at Cambridge, published findings in 2025 after accumulating observation runs across multiple transit events. The signal is statistically significant. It has not gone away with more data. It has gotten stronger. The planet itself is strange in exactly the right ways. K2-18b sits in the habitable zone of its star, a red dwarf called K2-18 in the constellation Leo. The planet is roughly 2.5 times Earth's diameter and about 8.5 times its mass, which puts it in a category astronomers call a sub-Neptune, or, depending on who you ask, a Hycean world. That last term was coined by Madhusudan's own group, a portmanteau of hydrogen and ocean. A planet with a hydrogen-rich atmosphere sitting above what could be a global liquid water ocean. That ocean, if it exists, would not look like ours. The pressure at depth would be extreme. The light filtering down through a hydrogen sky and 120 light-years of space would be dim and red. The star above, a red dwarf significantly cooler and smaller than the Sun, would appear perhaps twice the angular size of our Sun, but far less bright, glowing a deep amber rather than white-yellow. It is an alien ocean in every sense of the word. And something in that ocean, or at its surface, may be breathing out dimethyl sulfide into a sky that the James Webb Space Telescope can read like a paragraph. None of this is certain, but all of it is possible. And the fact that we can even ask the question with real data behind it is new. How you read the atmosphere of a planet you will never visit. The method is called transmission spectroscopy. When K2-18b crosses in front of its star, a transit event, a tiny fraction of the starlight passes through the planet's atmosphere before reaching us. Different molecules absorb different wavelengths of light. Carbon dioxide absorbs at one set of wavelengths, methane at another, dimethyl sulfide at another. The James Webb Space Telescope captures that absorbed spectrum with a precision no previous instrument could approach. What makes the James Webb different is not just its mirror, 6.5 meters across compared to Hubble's 2.4, but its infrared sensitivity and the fact that it operates in space, above the interference of Earth's own atmosphere. Previous telescopes gave us hints about exoplanet atmospheres. Webb gives us chemistry. The difference between hints and chemistry is the difference between seeing a figure in fog and actually reading their face. Madhusudan's team observed K2-18b across multiple transits and stacked the data. Carbon dioxide confirmed, methane confirmed, and then dimethyl sulfide, a tentative detection in earlier data that has strengthened with additional observations. Not a spike. A persistent, reproducible signal at the right wavelengths in a planet in the habitable zone, with a predicted liquid ocean beneath its atmosphere. Why this molecule specifically is the one that matters. Astronomers have a list of what they call biosignatures, molecules that, if found in a planetary atmosphere, would suggest the presence of life. Oxygen is on the list. So is ozone, so is nitrous oxide, and dimethyl sulfide. The reason dimethyl sulfide matters is specific and somewhat poetic. On Earth, It is produced primarily by phytoplankton and marine algae when they are grazed on or decompose. The sulfur compound inside them, dimethyl sulfoniopropionyate, breaks down into dimethyl sulfide, which volatilizes into the atmosphere. It is a byproduct of biological processes happening at the surface of the sea. It is one of the smells of ocean life. And it is not a molecule that forms easily through purely geological or photochemical processes, at least not in the quantities that Earth's biosphere produces. The key word there is at least. We do not know what K2-18b's chemistry is capable of. We do not have a full model of abiotic dimethyl sulfide production in a hydrogen-dominated atmosphere irradiated by a red dwarf. Some photochemical models suggest it is possible without life. Other models suggest the concentrations implied by the spectral signal would be extremely difficult to explain abiotically. The honest answer is that we do not yet know which models are right, and this uncertainty is not a failure of the discovery, it is exactly where science lives in real time. The history of false alarms and why this time feels different. In 1996, NASA held a press conference to announce possible signs of microbial life in a Martian meteorite called ALH84001. Structures that looked like fossilized bacteria, organic compounds, carbonate minerals associated with water. President Clinton made a public statement. 20 years of subsequent research found non-biological explanations for every single feature. The structures were too small to be bacteria by the standards of cellular biology. The organic compounds were likely terrestrial contamination. The carbonates could have formed at high temperatures incompatible with life. The excitement was real, and the caution was warranted, and the conclusion was ultimately disappointing. In 2020, a team led by Jane Greaves at Cardiff announced a detection of phosphine in the atmosphere of Venus. Phosphine is produced on Earth by biological or industrial processes. It was the kind of signal that makes people sit up straight. Then the data were reanalyzed by multiple independent groups. The signal turned out to be significantly weaker than originally reported and consistent with artifacts in the data processing. The authors revised their analysis. The debate continues, quieter now. K2-18b sits in that lineage. What makes it feel different is the instrument. The James Webb Space Telescope operates on a different level of sensitivity from anything used in previous biosignature searches. The spectral data from K2-18b is not marginal. It is the clearest atmospheric characterization of a habitable zone exoplanet yet achieved. The team is careful, and the molecule is present. Those two things coexisting matters. What a red dwarf star does to the question. About 80% of all stars in the Milky Way are red dwarfs. M-type stars, smaller, cooler, and dimmer than the Sun. If life can exist around red dwarfs, the universe is full of it. If it cannot, the search gets significantly harder. Red dwarfs are problematic for life in specific ways. They flare. They produce sudden, intense bursts of ultraviolet and X-ray radiation that can strip away planetary atmospheres over geological time. Planets in the habitable zones of red dwarfs are much closer to their stars than Earth is to the Sun, which means they are likely tidally locked, one face permanently in daylight, one permanently in night. The dynamics of a tidally locked world are strange. Some models show stable atmospheric circulation, carrying heat from the day side to the night side. Others show the night side freezing solid. The truth probably varies by planet. For K2-18b specifically, the red dwarf raises photochemical questions. The star emits a different spectrum from the Sun, far more red, far less ultraviolet. The atmospheric chemistry that occurs under red dwarf irradiation is not identical to what happens under a yellow star. Whether dimethyl sulfide can be produced abiotically under those specific conditions, at those specific pressures, in a hydrogen atmosphere, is exactly the kind of calculation that multiple groups are currently running. The answer is not in yet. The second example problem, why 2 is infinity. Right now, every piece of biology we understand comes from a single sample. All life on Earth descended from a common ancestor, one origin event, one biochemical framework, one version of how life works. We know about DNA and RNA and amino acids and cell membranes because that is what this one origin produced. We have no idea whether those features are universal requirements for life or local accidents of this particular chemistry experiment 4 billion years ago. A second example of life changes that completely. If microbes on K2-18b use the same amino acids, the same nucleic acid-base pairing, the same membrane structures, then those features are probably deep requirements, not accidents. If K2-18b life uses a different biochemistry entirely, then life is more flexible and more inventive than we imagined. Either result is revolutionary. One sample is an anecdote. 2 samples is the beginning of a science.
Astrobiologists have been saying this for decades in the abstract. Now there is a planet with a potential signal, real data, a real instrument, and a team actively working to get more observations. The abstract has become concrete. The Fermi paradox and what microbial life would mean for it. If life is found on K2-18b, microbial life, simple life, life in an alien ocean under a hydrogen sky, the immediate emotional response in most people would be wonder. And that would be correct. But the rational consequence is more complicated and significantly darker. The Fermi paradox asks why, if intelligent life is common in the universe, we have seen no evidence of it. The galaxy is old enough for a civilization to have spread through it many times over at sublight speeds. We have been looking for signals for decades. Nothing. One resolution is called the Great Filter, a barrier somewhere between simple chemistry and spacefaring civilization that almost nothing crosses. The crucial question is whether the filter is behind us or ahead of us. If life is rare, if the origin of life itself is the filter, then we are lucky survivors of an incredibly improbable event, and the rest of the universe is probably empty. If life is common, if microbes arise wherever conditions allow, then the filter is ahead. Something almost always stops life from becoming intelligent, or from surviving long enough to communicate, or from wanting to communicate at all. Finding microbial life on K2-18b would be evidence that life arises easily, that it is a predictable outcome of chemistry given suitable conditions, which would mean the great filter lies ahead of us, which would mean the silence of the universe has a reason, and that reason is that civilizations almost always fail before they can be heard. The discovery of alien microbes would be the most wonderful thing humanity has ever found. It would also be the most frightening. What the next few years actually look like. More observations of K2-18b are already planned. The James Webb Space Telescope will accumulate more transit data, and the dimethyl sulfide signal will either strengthen further or reveal itself as an artifact. Independent teams at other institutions will attempt to replicate the analysis with the same publicly available data and different methodology. The extremely large telescope, currently under construction in Chile, expected operational in the late 2020s, will add ground-based near-infrared capability that complements Webb's space-based observations. Simultaneously, photochemists and atmospheric modelers are working on the abiotic production question. Can K2-18b's chemistry produce dimethyl sulfide without life at concentrations matching the observed signal? Some preliminary models say no. Others are more ambiguous. This is not a question that will be resolved by telescope time alone. It requires theoretical chemistry work that is being done right now, published in preprints, argued about at conferences. The realistic timeline for a high-confidence answer
Either confirmation that the signal is robust and biogenic in origin, or an explanation that removes the need for biology, is probably years. Not decades. Years. The data are good enough that the question is answerable. We're just not at the answer yet. Living inside the question. There is something specific about this moment that deserves attention. We are not in the state of not knowing whether alien life exists. We are in the state of having a real signal from a real planet that has not been explained away and has gotten stronger with more data. That is a different epistemic position than any human being has ever occupied before. Previous generations lived with the question as philosophy. Does life exist beyond Earth? It was theology, speculation, science fiction. The stuff of Carl Sagan monologues and Drake equation calculations. Beautiful abstractions. But there was no data to point at. Now there is data. It may be wrong. It probably needs qualification. But it exists. A planet 120 light years away has something in its atmosphere that, on Earth, comes from living things. The signal has been measured multiple times by the most sensitive space telescope ever built. A team of rigorous scientists has published it in peer-reviewed literature after extensive analysis. The next step is not to wait and hope. It is to observe more, model more, challenge the interpretation more aggressively. We are one generation into the era of actually being able to ask this question with instruments capable of answering it. K2-18b might be the first whisper of an answer, or it might be a false alarm that teaches us something crucial about our methods. Either outcome advances the search. Either outcome changes what comes next. But the whisper is there, in a spreadsheet, in a spectrum, in a molecule that smells like the sea. The molecule is not the only thing that is strange about K2-18b. The strangeness goes deeper, into the architecture of the planet itself, and the more you look at it, the more the coincidences accumulate. Hycean worlds, Madhusudan's category, hydrogen plus ocean, were not taken seriously by most astrobiologists until recently. The assumption in the field for decades was that habitable worlds would look like Earth, rocky, with a nitrogen-oxygen atmosphere, with continents and shallow seas, and sunlight that photosynthesizing organisms could use directly. That assumption shaped what telescopes looked for, what models predicted, what counted as a promising target. K2-18b does not fit the assumption. It never did, and now it has a signal. The uncomfortable implication is that the universe may not care about our assumptions. Life, if it exists elsewhere, did not read our textbooks on what constitutes a habitable environment. It evolved, or did not evolve, in whatever chemistry happened to be available. Hydrogen-rich atmospheres covering global oceans are not rare in the galaxy. They may be among the most common planetary configurations that exist in habitable zones. If life can start in those conditions, the universe is far more populated than the Earth-centric models suggested. The chemistry of a Hycean ocean, what is actually down there. If K2-18b has a global ocean beneath its hydrogen atmosphere, that ocean is unlike anything on Earth in important ways, and possibly similar in others. The pressure at depth would be extreme. For reference, the deepest point in Earth's oceans, the Challenger Deep in the Mariana Trench, about 11 kilometers down, sits under a pressure of roughly 1,100 atmospheres. Life exists there. Microbes, fish, crustaceans, organisms that have been evolving under those conditions for tens of millions of years. The pressure on K2-18b's ocean would exceed Earth's deepest trenches at relatively shallow depths, given the planet's higher gravity. Temperature is the more complicated question. The surface of the ocean, if one exists, would be warmed by the star and by whatever greenhouse effect the hydrogen atmosphere provides. Deeper layers would be insulated. Some models predict temperatures compatible with liquid water throughout much of the water column. Others predict a transition at depth to high-pressure ice phases, exotic forms of water that are solid despite the heat because the pressure is so extreme. Life would be constrained to the upper layers if this is correct. If not, it could potentially exist throughout.
The chemistry of the ocean itself, what minerals are dissolved in it, what the pH is, what energy sources are available, depends on what lies beneath.
Does K2-18b have a rocky mantle, generating hydrothermal vents that pump chemical energy into the water? We do not know. We cannot see through the atmosphere and ocean to the interior with current technology. The composition of the sea floor, if there is one, is entirely unknown. What life in that ocean would need? Life, as we understand it, requires energy, a solvent for chemistry, and building blocks: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. K2-18b's ocean, if it exists, likely has hydrogen in abundance. Carbon and oxygen are present in the detected atmosphere. CO2 has been confirmed. The star provides energy, at least at the surface of the ocean. The energy problem on a Haitian world is not trivial.
Photosynthesis, the dominant energy-gathering strategy on Earth's surface, requires light to penetrate to the organisms doing the work. Under a thick hydrogen atmosphere, some light reaches the ocean's surface, but the spectrum is shifted toward infrared compared to what Earth's photosynthesizers evolved to use. Whether life could evolve photosynthetic machinery tuned to that spectrum is unknown. Chemosynthesis is an alternative. On Earth, ecosystems around hydrothermal vents at the ocean floor run entirely on chemical energy. Hydrogen sulfide and other compounds from the vents feed microbes that then feed everything above them in the food chain. Those ecosystems are independent of sunlight. If K2-18b has active geology generating hydrothermal activity, life could exist without photosynthesis entirely. The dimethyl sulfide signal, if biogenic, does not tell us whether the organisms producing it live at the surface or deep in a vent ecosystem.
It only tells us the molecule reached the atmosphere somehow. The red dwarf flare problem, and why it might not matter. Red dwarf stars flare. They produce sudden, intense bursts of ultraviolet and X-ray radiation, far more energetically than our Sun on a per-star mass basis. A major flare from a red dwarf can irradiate a nearby planet with enough energy to strip hydrogen from the upper atmosphere, generate harmful radiation at the surface, and disrupt any photochemistry that life depends on. For a long time, this was considered a major strike against life around red dwarfs. K2-18 is relatively quiet for a red dwarf, which is part of why its planet became a priority target. But the flare concern is not entirely resolved, and a Haitian world introduces a specific mitigation: the ocean. Life in a Haitian ocean would be protected from stellar radiation by the depth of the water above it. Water is an excellent absorber of ultraviolet radiation. Organisms living even a few meters below the surface would be largely shielded from flare events. This is another version of the same logic that makes Earth's deep sea a viable habitat, despite the enormous distances from sunlight and warmth. The atmosphere itself may also help. A hydrogen-rich atmosphere, depending on its composition and opacity, would absorb some of the incoming radiation before it reached the ocean's surface. The details depend on what else is mixed into that atmosphere, and those details are exactly what the James Webb Space Telescope is measuring. What we are actually detecting, the signal in context. It is worth being precise about what has been observed, because the precision matters for evaluating the claim. The James Webb Space Telescope observed K2-18b transiting its star multiple times. During each transit, the telescope collected the transmission spectrum, the pattern of wavelengths absorbed by the planet's atmosphere. The combined spectrum shows features consistent with carbon dioxide, methane, and dimethyl sulfide. The CO2 and methane detections are considered robust. The dimethyl sulfide detection is at a lower significance level. The current data puts it at roughly 2 to 3 sigma, which, in statistics, means the probability that it is a random fluctuation is somewhere between 2 and 5%. 2 to 3 sigma is not a discovery by the standards of physics, where 5 sigma is the conventional threshold. But in atmospheric spectroscopy of exoplanets, where the signals are tiny and the noise sources are complex, 2 to 3 sigma with a physically meaningful molecule in a habitable zone planet with a predicted ocean is taken seriously. The community's response to the paper has been cautious and engaged rather than dismissive, which, for a claim of this magnitude, is itself notable. More observations will push the significance in one direction or the other. If the signal is real, it should grow stronger with more data. If it is an artifact of noise or instrumental systematics, it should not reproduce consistently across independent observation runs. The people doing the work. Niku Madhusudan has been working on K2-18b for years. His group developed the Haitian world concept and has been among the most consistent voices arguing that the standard habitable world framework is too narrow. He is not a sensationalist. His papers are dense with caveats and error bars and careful delineations of what the data can and cannot support. When he says dimethyl sulfide is detected, he also says the abiotic alternative has not been ruled out. Both things are in the same sentence. That combination, genuine signal, genuine uncertainty, genuine care, is what distinguishes this from previous episodes of premature excitement. The claim is not that alien life has been found. The claim is that a molecule associated with life has been detected in an environment where life is theoretically possible, by an instrument capable of the measurement, by a team that has thought carefully about the alternatives. Other groups at other institutions are analyzing the same publicly released data. Some have found the dimethyl sulfide signal. Some have found it to be more marginal. This spread of results is not alarming. It reflects the genuine difficulty of the measurement and the fact that different analysis pipelines make different choices about noise treatment and baseline subtraction. The spread will narrow as more data accumulates. The other biosignature candidates, what else is being searched for? Dimethyl sulfide is not the only molecule on the biosignature candidate list. The broader search is running in parallel across dozens of planetary systems. Oxygen remains the most famous biosignature. On Earth, essentially all atmospheric oxygen is produced by photosynthesis. It would disappear within a few million years if life stopped producing it. Finding oxygen in a planetary atmosphere would be extraordinary evidence for biology. But it is harder to detect than molecules like dimethyl sulfide using current instruments, and it has known abiotic sources. Photolysis of water vapor can produce oxygen without biology. The detection standard for oxygen would need to include context. Oxygen combined with methane, for example, which react with each other and cannot coexist for long without constant replenishment, would be a much stronger joint signal. Nitrous oxide is another candidate. On Earth, it is produced by bacterial processes in soil and water. It has fewer known abiotic sources than oxygen. If the James Webb Space Telescope found nitrous oxide in a habitable zone atmosphere, it would attract serious attention. Phosphine, despite the Venus controversy, remains on the list for certain types of planetary environments. On gas giants like Jupiter and Saturn, phosphine forms through high-temperature chemistry in the deep atmosphere and gets dredged upward. On a rocky or ocean world, it would require biology or industrial processes. The list is long, and the James Webb Space Telescope is working through it systematically. K2-18b is not the only target. It is currently the most compelling one. What the silence of other planets in the sample means. The James Webb Space Telescope has been operating for a few years now and has characterized the atmospheres of multiple exoplanets. Most of them show no biosignature candidates. Most of them show chemistry consistent with geology and photochemistry alone. That is expected. Not every planet should have life. But the absence of signals in most of the characterized atmospheres is useful data. It constrains models of how common life-friendly conditions are. It suggests that whatever chemistry produced the K2-18b signal is not trivially reproduced on every similar planet. which is either evidence that the signal is unusual and therefore interesting, or evidence that the chemistry requires specific conditions that are not universal. One of the underappreciated aspects of the current moment is that the James Webb Space Telescope is not just searching for biosignatures on one planet. It is building a comparative dataset of exoplanet atmospheres. Over time, that dataset will allow scientists to ask statistical questions. What fraction of habitable zone planets show these signals? Are the signals correlated with other planetary properties? Is K2-18b an outlier or a representative? Those questions cannot be answered yet, but they will be answerable. The dataset is being built right now. The philosophical weight of waiting. There is a specific texture to this moment that is worth describing because it is historically unusual. For most of human history, the question of whether life exists beyond Earth was answered by faith, speculation, or silence. Copernicus removed Earth from the center of the solar system, but did not tell us whether other worlds were inhabited. Bruno speculated that they were, and was burned at the stake partly for suggesting it. The invention of the telescope opened other worlds to observation, but showed them as surfaces and atmospheres, not signs of life. The 20th century brought radio telescopes and SETI and the Drake equation, all ways of formalizing the search without yet having the instruments to conduct it meaningfully at the level of atmospheric chemistry. Now the instruments exist. They are operating. They are producing data. The data contains a signal that is not yet explained. We are not waiting for technology we do not have. We are waiting for more data from a telescope that is already in space, already pointing at this planet, already collecting photons. The answer, whatever it turns out to be, is coming. Not in generations, in years. That is a genuinely new situation for the species. The question has been abstract for so long that it is hard to process that it might not be abstract for much longer. The data is in transit, literally, light from K2-18b taking 120 years to reach Webb's mirror, Webb's electronics converting it to numbers, those numbers flowing to servers on Earth where people are reading them and arguing about what they mean. The universe, looking back. One of the stranger aspects of this discovery, if it holds, is the direction of looking. When we detect a biosignature on K2-18b, we are not receiving a message. The organisms producing dimethyl sulfide in that ocean, if they exist, have no knowledge of us. They are not signaling. They are just living, producing their metabolic byproducts into an atmosphere, as Earth's phytoplankton has been doing for 2.5 billion years without any interest in being detected. We are the observers. We built a telescope, put it in space, pointed it at a specific star, measured the light filtering through a specific atmosphere during a specific geometric alignment, and found something that we recognize from our own biology. The recognition is entirely on our side. The universe is not announcing itself. We are reading it the way you read footprints, not because the foot wanted to be found, but because the ground recorded the passing. If those organisms exist, they have been there for millions of years, or billions, in an ocean that has never been observed by any external intelligence. They have been producing dimethyl sulfide since long before Earth had complex life, long before our planet had oxygen, long before there were animals. They have been living in their hydrogen-dark ocean, entirely unaware of the pale yellow star 120 light-years away where, eventually, a species of upright primates would build instruments precise enough to read the chemistry of their atmosphere. The footprints are in the data. We are just now learning how to read them. The strangest part of the K2-18b situation is not the signal. It is how long the signal has been possible to detect, and how recently we built something capable of detecting it. The planet has been there for billions of years. The molecule, if it is there, has been drifting into the atmosphere for as long as whatever produces it has been alive. The star has been emitting light carrying the spectral fingerprint of that atmosphere for the entire duration of its existence. That light has been passing through this corner of the galaxy continuously, in all directions, including in the direction of Earth, for an unimaginable span of time.
For most of that time, Earth had no life capable of registering it. For most of the time that Earth had life capable of interest, there were no instruments. For most of the time that instruments existed, they were not sensitive enough. The James Webb Space Telescope reached full operation in mid-2022. The K2-18b observations began shortly after. We are 3 years into a detection that the universe has been broadcasting for potentially billions of years. There is something vertiginous about that. The message, if it is a message, if biology is the source, was not sent to us. It was not encoded for our detection. It has been sitting in the light this whole time, waiting for a receiver sensitive enough to notice. We just built one. The comparative planetology angle, what the rest of the Webb sample tells us. The James Webb Space Telescope has not only been looking at K2-18b. Its exoplanet program covers a range of targets: hot Jupiters, warm Neptunes, rocky planets, sub-Neptunes, and it has been characterizing their atmospheres since 2022 with unprecedented clarity. TRAPPIST-1 is one of the most watched systems. 7 Earth-sized planets orbit an ultra-cool red dwarf just 40 light-years away. 3 of them sit within or near the habitable zone. Webb has already observed several TRAPPIST planets and found, for the most part, no significant atmospheres, or at least no dense atmospheres detectable in transit. TRAPPIST-1b and TRAPPIST-1c both show results consistent with bare rock or very thin atmospheres. This is not entirely surprising. Ultra-cool red dwarfs are extremely active early in their lifetimes and may strip away planetary atmospheres entirely before the planets could develop life. The TRAPPIST null results are important context for K2-18b. They show that not every planet in a habitable zone retains its atmosphere. K2-18b has a substantial, clearly detectable atmosphere. That alone makes it unusual among the characterized sample. Add the habitable zone location, the predicted ocean, and the biosignature candidate, and the coincidence of favorable factors becomes striking enough to demand explanation. Whether the explanation requires biology remains open, but the comparison makes K2-18b look less like a random detection and more like a genuinely anomalous object. The sulfur chemistry question: what abiotic chemistry can and cannot explain? The critical, unresolved scientific question right now is specific. Can K2-18b's chemistry produce dimethyl sulfide at the atmospheric concentrations implied by the spectral signal without any biological input? This is not a simple question. Dimethyl sulfide photochemistry in hydrogen-dominated atmospheres is a relatively understudied area. Most of the atmospheric chemistry models developed over the past few decades were built for nitrogen-oxygen atmospheres like Earth's, or for the hydrogen-helium envelopes of giant planets. A hydrogen atmosphere with water vapor, carbon dioxide, methane, and potentially liquid water below it is a specific regime that is only now being modeled in detail. Several groups have published preprints attempting to address this. The general finding, with significant variation across different modeling assumptions, is that abiotic production pathways for dimethyl sulfide in Hycean world conditions are limited, but not entirely excluded. One preliminary study from a group at Johns Hopkins suggested that under certain photochemical conditions, dimethyl sulfide could accumulate in the upper atmosphere of a Hycean world at detectable concentrations without biology. Another analysis from a group at MIT found that the steady-state concentrations required to match the observed signal were orders of magnitude above what abiotic pathways could plausibly produce. Both cannot be right. The disagreement reflects genuine uncertainty about the inputs: the UV flux from K2-18, the depth of the ocean, the surface chemistry, the rate constants for key reactions at non-Earth pressures and temperatures.
Resolving it requires more precise spectral data, more refined planetary models, and more laboratory measurements of the relevant reaction rates under simulated Hessian conditions. All of that work is ongoing. What laboratory astrophysics has to do with it? One of the underappreciated requirements for interpreting the K2-18b data is laboratory work on Earth, measuring the spectroscopic properties of molecules under conditions that mimic the planetary atmosphere, measuring the rates of chemical reactions that produce and destroy dimethyl sulfide under non-Earth pressures and temperatures, and checking whether the spectral features attributed to dimethyl sulfide in the Webb data might overlap with features from other, more exotic molecules we have not considered. This last point is not trivial. The spectral resolution of the James Webb Space Telescope, while extraordinary, is not infinite. A spectral feature at a particular wavelength could in principle come from multiple different molecules whose absorption features overlap in that region. Confirming that the feature is specifically from dimethyl sulfide rather than from some other sulfur compound or some unexpected atmospheric constituent requires high-precision laboratory spectra of candidate molecules measured at the relevant temperatures and pressures. Several laboratory groups are currently generating exactly this kind of data. The Molecular Astrophysics Group at NASA Ames, groups at the HITRAN Database Consortium, and several European research teams are producing updated spectral line lists, essentially the reference catalogues that tell you what wavelengths each molecule absorbs at, for molecules relevant to Hessian world atmospheres. Those catalogues are the Rosetta Stone between the telescope data and the molecular identification. If the laboratory measurements are incomplete or inaccurate, the interpretation of the telescope data can be wrong, even if the telescope data itself is perfect. This is one of the quieter but more important fronts of the current work.
Not glamorous.
Essential. The statistical argument. How many chances does life get? One way to think about the K2-18b situation is probabilistically. How likely is it that we looked at one of the first habitable zone exoplanets we could characterize in detail and found a biosignature candidate by chance? The James Webb Space Telescope has characterized the atmospheres of roughly a few dozen exoplanets in meaningful detail since 2022. Of those, a small number sit in or near habitable zones with predicted liquid water. K2-18b is the most favorable among those, the most clearly habitable zone, with the best atmospheric signal-to-noise, with the predicted ocean. It is the best candidate in the sample almost by definition, because it was prioritized precisely for those reasons. The fact that the best candidate in a carefully selected sample shows a biosignature candidate is not the same as a random discovery. The target was chosen because it had the best chance of showing something interesting. Finding something interesting in the most promising target is the expected outcome if interesting things are to be found anywhere, and also expected if the observer is subject to confirmation bias, or if the instruments produce artifacts preferentially in high-signal targets. None of that makes the detection wrong, but it is relevant to the prior probability assessment. The team did not randomly point the telescope at the sky and find dimethyl sulfide. They pointed it at the most favorable known target and found dimethyl sulfide. The significance of that depends on assumptions about how common biosignature planets are, which is exactly what is being debated. The carbon silicate cycle and why K2-18b might be stable. One of the challenges for life on a planet orbiting a red dwarf is geological stability over the timescales required for evolution to do meaningful work. Earth's carbon silicate cycle, the slow geological thermostat that regulates CO2 over millions of years through weathering and volcanism, requires a rocky mantle, tectonic activity, and an interface between rock and atmosphere or ocean where the chemistry can happen. K2-18b's interior is uncertain, but some models suggest it has a rocky mantle beneath the ocean layer. If so, and if that mantle is active, then a version of the carbon silicate cycle could operate at the ocean-rock interface. That would provide long-term climate stability,
a mechanism for keeping the planet habitable over the billions of years that complex life on Earth required. There is also the question of how long K2-18b has had its current configuration. Red dwarfs are long-lived. K2-18 is estimated to be several billion years old. If the planet has been in something like its current state for most of that time, life has had as much time to develop as it has had on Earth. Not more, but not less.
The time argument against life around red dwarfs, that early stellar activity prevents life from getting started, is most damaging in the first billion years. After that, if life established itself, the star settles into a quieter phase that could sustain it. The origin of life problem and why K2-18b makes it sharper. Even if K2-18b has all the conditions for life, liquid water, chemical energy, organic molecules, geological stability, life still has to start. And the origin of life is one of the genuinely unsolved problems in science, not in the sense that we are making no progress, but in the sense that we do not yet have a widely accepted, complete account of how chemistry became biology. The leading frameworks, hydrothermal vent chemistry, RNA world scenarios, prebiotic chemistry on mineral surfaces, all have experimental support for pieces of the story and significant gaps in others. The transition from complex prebiotic chemistry to self-replicating, membrane-enclosed, metabolizing systems has not been reproduced in a laboratory from scratch. The number of steps required, the probability of each step, the range of conditions over which each step can occur, all of these are active research areas with significant uncertainty. If life is found on K2-18b, the origin of life question becomes dramatically sharper. If life started independently in a Haitian ocean environment that is substantially different from any of the Earth environments proposed as origin of life sites, then either life can start through multiple different chemical routes, which would suggest it is a robust and common outcome of chemistry, or there is something universal about the chemistry of life's origin that works in radically different environments. Either conclusion would be transformative for the field.
If K2-18b turns out to have no life despite favorable conditions, that is also informative. It suggests that the origin of life is not automatic, even when conditions are suitable, which constrains how common life can be. What happens to the search after K2-18b? Whatever the K2-18b data ultimately shows, it has already changed the search in one concrete way. It has demonstrated that the James Webb Space Telescope can detect atmospheric biosignature candidates in habitable zone exoplanets. The method works at the level of sensitivity required. That was not guaranteed before the K2-18b results. The next generation of targets is already being assembled. The JWST Cycle 3 and Cycle 4 programs include additional observations of K2-18b and of other Haitian world candidates. The Nancy Grace Roman Space Telescope, expected to launch in the late 2020s, will conduct wide-field surveys that could identify additional habitable zone planets in nearby systems.
The Habitable Worlds Observatory, a proposed NASA flagship mission concept, is designed specifically for the direct imaging and spectroscopy of Earth-like planets around Sun-like stars, with a target sensitivity sufficient to detect oxygen and water vapor simultaneously. Its proposed launch window is somewhere in the late 2030s or early 2040s. If K2-18b's signal is confirmed as biogenic over the next few years, the pressure to accelerate those programs will be immense. A confirmed biosignature on one planet changes the political and funding calculus of space science in ways that are difficult to predict but easy to imagine. Every question about budget allocation for space telescopes becomes a question about whether we are going to follow up on the most important discovery in human history. The communication problem, what we could never say to them. There is a thought that is difficult to avoid when looking at the K2-18b data,
even though it is not science. If there is life in that ocean, microbial life, nothing remotely intelligent, just chemistry that learned to replicate and metabolize in a hydrogen-dark sea, we can never tell it that we found it. The communication problem is not just the distance, though 120 light-years makes two-way communication over human timescales impossible. The problem is more fundamental. We have nothing to say to a microbe. It would not receive our signal. It would not interpret our signal. It would not know that anything had changed. It lives in its ocean exactly as it lived before we built a telescope, and after, and regardless. Our discovery of it changes nothing in its world. What changes is in our world. The category of inhabited planet expands from 1 to 2. The question, are we alone, shifts from having no empirical answer to having a data-based candidate answer. The self-understanding of the species shifts, subtly, unevenly, incompletely, but shifts. Some people will feel the pull of that shift immediately. Others will process it over years. Some will resist it for reasons that have nothing to do with science. But the shift will happen, because the data will accumulate, and the accumulation will be undeniable. The timing of this, why now and not earlier or later? The James Webb Space Telescope was proposed in 1996, the same year as the ALH 84001 press conference. It was approved for development in 2003. It launched in December 2021, 25 years after conception. It reached its operational orbit, the second Sun-Earth Lagrange point, in January 2022. First science images were released in July 2022. The K2-18b observations began that year and have continued since. The history of the telescope's development includes budget overruns that nearly cancelled it, technical challenges that took years to resolve, political fights about science funding that endangered it multiple times. The fact that it launched at all is not obvious in retrospect. Different decisions at different moments could have resulted in a different telescope, or no telescope, or a telescope operating a decade later. The timing of the K2-18b discovery, if it holds, is a contingent fact about the specific history of a space agency, a budget process, and a set of engineering decisions made by specific people over 25 years. In another timeline, the telescope launched in 2012, and the K2-18b data was published in 2014. In another, it launched in 2030, and we are still waiting. We happen to be in the timeline where it launched when it did, and the first compelling biosignature candidate appeared in 2025. That contingency does not make the science less real, but it is a reminder that discoveries are not inevitable. They happen when the instruments and the targets and the moment align. The thing nobody says out loud in the papers, but that everyone is thinking, is what it would mean for religion. Science has been here before. The Copernican displacement removed Earth from the center of the cosmos and took several generations to fully metabolize. The Darwinian displacement removed humans from a special biological category and is still being metabolized in some quarters. A confirmed discovery of life beyond Earth would be the third displacement in the series, and unlike the first two, it would be empirical in a way that is much harder to reinterpret metaphorically. You can argue about the theological implications of a heliocentric solar system. It is considerably harder to argue with a spectrum. The theological responses to a confirmed biosignature are varied and have been discussed in academic literature for decades, long before the K2-18b data existed. Some traditions find extraterrestrial life compatible with their cosmology. A universe full of life is a universe that more fully expresses divine creativity. Others find it challenging. Doctrines of special human status, unique redemption, singular creation all sit uneasily with a cosmos teeming with biology. The specific challenge depends on the specific tradition, the specific doctrine, and the specific kind of life found. Microbial life in an alien ocean is a different theological challenge than an intelligent civilization. What is notable is that the conversation has already started.
not in response to the K2-18b data, but in anticipation of it. Several Vatican-affiliated astronomers have been actively engaged in astrobiology for years. Protestant and Jewish theologians have published on the implications.
Islamic scholars have weighed in. The intellectual preparation has been happening. Whether it has happened broadly enough or deeply enough to absorb a confirmed discovery without social disruption is a different question. The problem of what counts as confirmation. One of the subtler difficulties in the K2-18b situation is that there is no agreed-upon standard for what would constitute confirmed evidence of extraterrestrial life. This is not a new problem. It has been discussed in astrobiology for decades, but it becomes acute when a real signal is on the table. In particle physics, a 5 sigma threshold is the conventional standard for discovery. That means the probability that the signal is a statistical fluctuation is less than 1 in 3.5 million. The reason for that threshold is specific to particle physics. The experiments can be repeated under controlled conditions, the theoretical models are precise, and the systematic errors are well understood. 5 sigma in a particle detector means something definitive. In exoplanet atmospheric spectroscopy, the situation is different. The signal is not from a controlled experiment. It is from a natural system that cannot be replicated in a laboratory. The theoretical models of planetary atmospheres, of atmospheric chemistry, of possible abiotic production pathways have uncertainties that are much larger than in particle physics. 5 sigma in a transmission spectrum does not mean the same thing as 5 sigma in a collider experiment. The community has been working toward what Sarah Seeger at MIT calls an agnostic biosignature framework, a set of criteria that do not assume any particular biochemistry, that combine multiple independent lines of evidence, and that explicitly account for the uncertainty in abiotic alternatives.
Under such a framework, confirmation of life on K2-18b would require not just a robust dimethyl sulfide signal, but also the absence of plausible abiotic explanations, corroborating signals from other molecules, and independent replication by multiple telescopes and analysis teams. None of that exists yet. The data is currently at the beginning of that process, not the end. What the exoplanet community is actually arguing about right now, the genuine scientific debate, is not whether the dimethyl sulfide signal exists. Most people who have looked at the data agree there is something there. The debate is about interpretation and about what additional data is needed. One camp argues that the priority should be more observations of K2-18b with the James Webb Space Telescope, accumulating more transit data to push the dimethyl sulfide signal to higher significance before investing in theoretical modeling of abiotic alternatives. More data first, more analysis second. This camp worries that premature theoretical work will prematurely close off the biological interpretation by finding plausible abiotic explanations that are not actually correct. Another camp argues that the priority should be parallel investment in atmospheric chemistry modeling and laboratory spectroscopy, building the theoretical framework needed to distinguish biological from abiotic signals before claiming that the distinction has been made. More theory first, more telescope time second. This camp worries that accumulating telescope time on a planet whose signal cannot yet be interpreted unambiguously is inefficient and risks misleading the public. A third group is focused on widening the sample, using the time that would otherwise go to more K2-18b observations to characterize other Hycean world candidates. If K2-18b is not unique, finding similar signals in other planets would be far more compelling evidence for biology than any amount of additional data on a single planet. Because independent occurrences of the same biosignature on different worlds would be very difficult to explain without life. All three camps have legitimate scientific arguments. The telescope time allocation committees at NASA and ESA are making decisions between them right now. The scale problem.
why 120 light-years is both close and unreachable. 120 light-years sounds like a fact about astronomy. It is also a fact about the human condition. Light travels at roughly 300,000 kilometers per second, about 9.5 trillion kilometers per year. 120 light-years is approximately 140 trillion kilometers. The fastest human-made object ever launched is the Parker Solar Probe, which reached speeds of around 690,000 kilometers per hour at its closest approach to the Sun. At that speed, which is not a cruise speed, it is a brief peak during a gravitational slingshot, reaching K2-18b would take about 190,000 years. No technology on any foreseeable engineering roadmap changes that number by more than a factor of maybe 10 or 20. Even the most optimistic laser sail concepts, like the Breakthrough Starshot project aimed at Proxima Centauri, 4.2 light-years away, project speeds of about 20% of the speed of light with a travel time of roughly 20 years. K2-18b is 28 times farther than Proxima Centauri. At the same speed, the travel time is about 560 years. We are not going there. Not with any technology that physics currently permits. The organisms in that ocean, if they exist, will never be visited in any human lifetime, or in any lifetime that maps onto current human civilization. What we have is light. The signal in the light. The chemistry in the signal. That is everything we will ever have, unless physics reveals a door we do not currently know exists. What the signal tells us about the ocean, reading between the lines, the dimethyl sulfide in K2-18b's atmosphere, if biogenic, carries indirect information about the ocean beneath. On Earth, dimethyl sulfide production is concentrated in the upper ocean, the photic zone, roughly the top 200 meters where sunlight penetrates. That is where phytoplankton live, where the biological cycle of dimethyl sulfonio-propionate synthesis and breakdown occurs, where dimethyl sulfide volatilizes into the atmosphere. The rate of production scales with the productivity of the surface ocean, which scales with temperature, nutrient availability, and light. If the same logic applies to K2-18b, which is an assumption, not a certainty, then a detectable dimethyl sulfide signal implies a productive surface ocean, not a dead ocean with a few isolated organisms, an ocean with enough biological activity at or near the surface to maintain a steady flux of the molecule into the atmosphere against whatever photochemical destruction processes are removing it. That is not a small amount of life. A productive ocean on K2-18b, by analogy with Earth, would mean something like the biological productivity of the modern Earth ocean. Billions of organisms per liter of surface water, producing and consuming organic matter on timescales of days to weeks. If the analogy holds, which it may not. The biochemistry could be entirely different, producing dimethyl sulfide through pathways that operate at very different concentrations of organisms. The point is that a detectable atmospheric signal implies not just the existence of life, but life at a scale sufficient to maintain a global atmospheric fingerprint. That is a meaningful statement about the system, not just a detection of a trace. The role of sulfur in the history of life on Earth. Sulfur is the sixth most abundant element in living organisms on Earth, after oxygen, carbon, hydrogen, nitrogen, and phosphorus. It appears in two amino acids, cysteine and methionine, that are essential for protein folding and function. It appears in coenzyme A, which is central to metabolism. It appears in iron-sulfur clusters, some of the oldest and most fundamental electron transfer systems in cellular biochemistry, systems that may date back to the very origin of life.
The metabolic cycling of sulfur was one of the first planetary biogeochemical cycles to operate on early Earth.
Sulfate-reducing bacteria, organisms that use sulfate as an electron acceptor in the same role that oxygen plays in aerobic respiration, appear in the fossil record more than 3.5 billion years ago. Long before oxygen, long before complex cells, sulfur metabolism is ancient. Dimethyl sulfide sits in the sulfur cycle. Its production from dimethyl sulfoniopropanate, its release into the atmosphere, its oxidation back to sulfate. These are steps in a global sulfur cycling process that has been running on Earth for billions of years. The fact that the biosignature candidate from K2-18b involves a sulfur compound is notable. Sulfur is a plausible early metabolic substrate on any world with liquid water and geological activity. If life elsewhere starts with sulfur-based metabolism, as some researchers believe early Earth life did, then sulfur-containing biosignature molecules are exactly what you might expect to find. The problem of Earth-centric biology.
Every biosignature on the current candidate list is derived from Earth biology. Dimethyl sulfide, Earth organisms produce it. Oxygen, Earth photosynthesis produces it. Methane combined with oxygen. Earth biology maintains both simultaneously. Phosphine, Earth bacteria produce it. The entire framework is built from the one example we have. This is unavoidable. We cannot build a framework from examples we do not have. But it creates a specific risk. Life on other worlds may produce molecules we have not thought to look for, may not produce the molecules we are looking for, or may produce them through abiotic chemistry that our Earth-centric intuition says should require biology. Some researchers are trying to build what they call agnostic biosignatures, signals that would indicate life without assuming Earth-like biochemistry.
One approach looks for molecules that are too thermodynamically unstable to accumulate to detectable levels without active replenishment. Life as the replenishment mechanism. Another approach looks for complexity itself, a complex mixture of organic molecules in a planetary atmosphere, with a chemical diversity that seems to exceed what simple abiotic chemistry should produce. These agnostic approaches are theoretically appealing but practically difficult. The James Webb Space Telescope cannot resolve individual molecules in a complex mixture the way a laboratory mass spectrometer can. It detects broad spectral features that may come from one molecule or from several overlapping ones. The resolution required for truly agnostic biosignature detection may require future technology beyond Webb. For now, the search is necessarily Earth-centric. The dimethyl sulfide detection is evaluated against Earth biochemistry because that is the only biochemistry we know. If K2-18b life uses different chemistry, we might be looking at the wrong wavelengths entirely. The psychological effect on the people doing the work. What is it like to be Niku Madhusudan right now, or any member of the team that published the K2-18b results? Scientists are trained to suppress the emotional register of their work in publications. Papers are written in passive voice, with hedged language, without first-person accounts of what it felt like to look at the data for the first time. The norms exist for good reasons. They prevent personal investment from distorting interpretation. They maintain the appearance of objectivity that scientific communication requires. But they also create a gap between the public presentation of science and the actual experience of doing it. Madhusudan has given interviews where he speaks with evident care about not overstating the findings. He uses phrases like, "if confirmed," and "requires further investigation," and "we cannot rule out abiotic explanations." That carefulness is not performance. It is genuine scientific practice and genuine awareness of what is at stake. Getting this wrong in either direction, either claiming confirmed life when it is not confirmed, or dismissing a real signal through overcaution, would have lasting consequences for the field and for public trust in science. Behind that carefulness, though, is a team of people who ran the analysis, saw the dimethyl sulfide feature in the spectrum,
and had to sit with what it might mean? Who went home that night knowing what was in the data? Who have been living with that knowledge while writing the papers, giving the talks, answering the emails, and waiting for more telescope time? The emotional texture of that experience, of holding a possible world-changing discovery at arm's length until you are sure, is not captured in the literature.
The pale blue dot, and what a second one would do to the image.
In 1990, the Voyager 1 spacecraft turned its camera back toward the inner solar system from a distance of about 6 billion kilometers. In the resulting photograph, Earth appears as a fraction of a pixel, a pale blue dot, as Carl Sagan described it, suspended in a sunbeam in the vastness of space. Sagan's meditation on that image became one of the most widely quoted passages in science writing. Our planet is a lonely speck in the great enveloping cosmic dark. In our obscurity, in all this vastness, there is no hint that help will come from elsewhere to save us from ourselves. The power of the image was its isolation, the only known inhabited world reduced to a point of light. If K2-18b's signal is confirmed as biogenic, there are two pale blue dots. The image does not literally change. K2-18b is too far away to photograph. But the conceptual image changes. The loneliness that made Sagan's meditation so powerful dissolves, at least partly. We are not the only speck. There is another, 120 light-years away, in a hydrogen ocean under a dim red star. Something is alive. And then the question immediately becomes, how many more? If 2, why not 20? Why not 200? Why not 200,000? The mathematics of a universe with inhabited worlds, rather than one inhabited world, starts to produce very large numbers very quickly. A galaxy with 1 trillion stars, and even a tiny fraction of them hosting life, is a galaxy full of biology. Most of it invisible to us, all of it indifferent to us, none of it in contact with us, but present, real, alive. The data from K2-18b has already changed one thing that cannot be unchanged. It has given the search a specific address. Before this, the search for extraterrestrial life was conducted against a background of generality. We were looking for biosignatures in the spectra of exoplanets in habitable zones. Plural, abstract, statistical. Now there is a specific planet with a specific signal at specific coordinates in the constellation Leo, 120 light-years away, that has been observed by a specific telescope, producing specific numbers that a specific team of scientists has published and that other scientists are now replicating and challenging. The search has an object. That object has an address. The address has data attached to it. This shift from general to specific is not just psychological. It concentrates scientific resources in a way that general searches cannot. Telescope time, computing time, laboratory work, theoretical modeling. All of it can now be directed at a single target with a known profile. The efficiency of targeted investigation exceeds the efficiency of surveys by orders of magnitude. Every hour of James Webb time on K2-18b produces information directly relevant to the specific question. Is the dimethyl sulfide signal biogenic? Every hour on a random survey produces information that may or may not bear on any question of comparable importance. The scientific community knows this. The allocation committees know this. The next several years of exoplanet atmospheric spectroscopy will be shaped by what the K2-18b data has established. Not because the discovery is confirmed, but because the candidate is real enough to demand investigation. The ocean beneath the signal. Physical constraints. If K2-18b has a Hycean ocean, its properties are constrained by the planet's mass and radius, and the atmospheric composition the James Webb Space Telescope has measured. The ocean is not arbitrary. Physics determines what it can be. The planet's radius is roughly 2.5 times Earth's. Its mass is about 8.5 times Earth's.
Those two numbers together constrain the interior structure. The relationship between radius and mass tells you something about the density and therefore the composition. A planet of that mass and radius could be explained by a rocky core with a thick hydrogen-water envelope, or by a denser planet with less hydrogen, or by several other configurations. The current best-fit models from Madhusudan's group favor the Hessian configuration: a rocky core, a high-pressure water layer potentially containing liquid water, and a hydrogen atmosphere above it. The water layer, if it exists, would be under pressures of perhaps thousands of atmospheres at depth. High-pressure water has phases, states of matter, that do not exist at Earth's surface conditions. Ice 7, Ice 10, superionic water. These are forms in which water molecules are organized differently from liquid water or ordinary ice.
Life, as we understand it, requires liquid water, a solvent in which molecules can diffuse, react, and organize. Whether life could exist at the boundary between liquid water and exotic high-pressure ice phases is unknown. There is no experimental data on biology at those pressures because nothing on Earth has evolved to live there. The upper ocean, near the base of the atmosphere, would be at lower pressures, perhaps a few hundred to a few thousand atmospheres, and possibly at temperatures compatible with liquid water chemistry. If life exists in K2-18b's ocean, it most plausibly lives in that upper region, which is also where dimethyl sulfide, if produced biologically, would most plausibly reach the atmosphere. The detection method and its intrinsic limitations. Transmission spectroscopy is powerful, but not omniscient. There are specific things it cannot tell us about K2-18b that would be important for confirming a biosignature. It cannot tell us the vertical distribution of molecules in the atmosphere, whether dimethyl sulfide is concentrated near the surface, near the ocean-atmosphere boundary, or distributed throughout the atmosphere. That matters for distinguishing biological sources from photochemical destruction products that might form at higher altitudes. It cannot tell us the absolute abundance of dimethyl sulfide, only its relative presence in the transmission spectrum. Translating the spectral signal into a parts per million or parts per billion concentration requires models of the atmospheric structure, temperature profile, and pressure, all of which have uncertainties that propagate into the abundance estimate. It cannot tell us anything about the surface. The transmission spectrum samples the limb of the planet, the thin ring of atmosphere visible during the transit, not the dayside or nightside surface. Whether the ocean below is liquid, frozen, or absent, whether there are volcanic features, whether the surface chemistry provides the feedstocks for biology, none of this is accessible through transmission spectroscopy. Future observing modes with the James Webb Space Telescope, specifically secondary eclipse spectroscopy, which measures the planet's emission as it passes behind the star, can provide complementary information about the dayside atmosphere. Reflected light spectroscopy, which would require a different kind of telescope, could eventually provide information about the surface. Both are on the roadmap. Neither is available yet for K2-18b. The question of microbial complexity, what kind of life might be there, assuming for a moment that the signal is biogenic, what kind of life are we talking about? The organisms producing dimethyl sulfide on Earth range from single-celled phytoplankton to bacteria to some algae. They are all microbial or close to it. Complex multicellular life, animals, plants, fungi, does not produce dimethyl sulfide in significant quantities. If K2-18b life is producing dimethyl sulfide through analogous biochemistry, it is most likely microbial. Microbial life on Earth has been present for roughly 3.5 to 4 billion years. Complex multicellular life, the kind that leaves recognizable macroscopic fossils, has been present for roughly 600 million years. The transition from microbial to complex took somewhere between 3 and 3.5 billion years and may have required very specific conditions,
a particular sequence of evolutionary innovations, specific atmospheric oxygen concentrations, possibly even a specific kind of geological event like the Cambrian explosion trigger. If K2-18b has been habitable for several billion years and hosts life, that life is likely microbial.
The conditions in a Haitian ocean, high pressure, limited light, hydrogen rather than oxygen as the dominant atmospheric gas, do not obviously favor the evolution of complex multicellular organisms. Complex life on Earth is aerobic. It runs on oxygen, which is produced by photosynthesis and accumulated in the atmosphere over about 2.5 billion years of biological activity. K2-18b's atmosphere contains methane and CO2, not oxygen. If that atmosphere reflects the biology, it reflects a world at an early evolutionary stage, metabolizing, cycling sulfur and carbon, but not yet photosynthesizing in a way that would oxygenate the world. Microbial life and complex life are not equivalent in their implications. Microbial life on K2-18b would confirm that biology is not a singular accident of Earth chemistry.
Complex life on K2-18b, if it could somehow be detected, which it currently cannot, would suggest that the evolutionary path from microbe to multicellular organism is not uniquely difficult. The distinction matters enormously for the Fermi paradox and for our sense of how common intelligence might be. Currently, we can only access the microbial question. The geological argument, what K2-18b's interior might be doing. Life requires energy. On a Haitian world without surface sunlight reaching deep enough for photosynthesis to be the dominant energy source, the most plausible energy supply is geological. Hydrothermal systems at the ocean floor, if there is an ocean floor, releasing chemical energy through the reaction of hot rock and water. Hydrothermal systems on Earth support ecosystems entirely independent of photosynthesis.
The Lost City hydrothermal field in the Atlantic Ocean, discovered in 2001, runs on the reaction between seawater and olivine-rich mantle rock, a process called serpentinization that produces hydrogen and heat without requiring volcanic activity in the conventional sense. The ecosystems at Lost City are dominated by archaea and bacteria that derive energy from hydrogen and methane rather than from sunlight. K2-18b, if it has a rocky mantle, may have serpentinization or conventional hydrothermal activity. The planet's age, K2-18, is estimated to be several billion years old, means it has had time for radioactive heat production in its interior to drive geological activity. Older planets generally have less geological activity as their radioactive isotopes decay and the interior cools. But several billion years is not necessarily enough time for complete geological quiescence in a planet of K2-18b's size. Larger planets cool more slowly. K2-18b's larger mass compared to Earth may mean it retains interior heat more effectively. If hydrothermal systems exist on the ocean floor of K2-18b, they provide both energy and a chemical interface between the rocky mantle and the ocean. That interface is where some origin of life researchers believe life began on Earth. The conditions are chemically similar wherever they occur, not because of biology, but because the chemistry of hot rock and water is determined by physics and geology that operates the same way across the universe. If life can start in those conditions on Earth, the argument that it could start in the same conditions on K2-18b is not unreasonable. The public communication challenge, how to say maybe about the biggest question. Science communicators, journalists, and scientists themselves face a specific challenge with the K2-18b story that is different from most scientific communication challenges. The challenge is not explaining a difficult concept.
The challenge is communicating meaningful uncertainty about an enormously significant possibility without either underselling it to the point of irrelevance or overselling it to the point of misleading.
Scientists may have found evidence of alien life, is the headline formulation. It captures the significance while preserving the uncertainty with that may. But it is also the kind of headline that readers have learned, from experience, to file under "this will probably turn out to be nothing." The history of biosignature false alarms, ALH 84001, phosphine on Venus, the WOW signal, various premature announcements, has created a skepticism in the public that is actually healthy, but that can impede the communication of genuine progress. The K2-18b situation is different in kind from most of those false alarms, for reasons that require some technical explanation to appreciate. That technical explanation does not fit in a headline. The gap between may have found evidence and the actual status of the data, statistically significant, not yet confirmed, biogenic interpretation, not ruled out, abiotic alternatives, not ruled in, requires paragraphs, not sentences. Most people do not read the paragraphs. The consequence is a public discourse about K2-18b that is polarized between excited overinterpretation and dismissive skepticism, with relatively few people occupying the correct position. This is the most significant biosignature candidate ever obtained, from the best instrument ever built, in a planet in the habitable zone with a predicted ocean, and it has not been explained away, and it needs more data, and we are getting more data, and the answer is coming in years. The search for life and the search for meaning. The hunt for extraterrestrial life is also, always, a hunt for a certain kind of meaning, a confirmation that the universe produces biology as a matter of course, that life is not a cosmic accident, but a cosmic fact, that the chemistry that runs in our cells is not a lonely local experiment, but a universal phenomenon. That search has a philosophical and emotional dimension that sits alongside the scientific one. When Carl Sagan argued for SETI funding, when Frank Drake designed his equation, when NASA included a plaque on Pioneer and a record on Voyager, these were scientific activities with a human need underneath them. The need to know that we are not alone, that consciousness and biology and the whole extraordinary fact of living things is not isolated to this one small world orbiting this one average star. K2-18b does not answer that need with certainty, but it is the closest thing to an answer that has ever existed in empirical form. A planet, a signal, a molecule. Data that has been measured and published and argued about and not explained away. That is not nothing. In a search that has run for decades with no confirmed result, a serious, unrefuted candidate is a qualitatively different thing from no candidate at all. Whether the candidate confirms is a question for the coming years. What it has already done is shift the search from a question about whether the universe could contain other life to a question about whether this specific planet does. That shift, from the general to the specific, from the philosophical to the empirical, is permanent, regardless of what the additional data shows. The silence and the signal simultaneously. There is a particular quality to this moment in the search for life that deserves sitting with rather than rushing past. We have a signal that might be life. We also still have the silence. No radio transmissions, no spacecraft, no megastructures, no undeniable detection of intelligence anywhere in the universe.
Both are true simultaneously.
A possible biosignature from one ocean world 120 light-years away, and complete silence everywhere else. That combination is consistent with a universe where microbial life is common and complex, or intelligent life is rare, which is one of the more philosophically striking outcomes. Life everywhere, intelligence almost nowhere. The universe producing biology with mechanical regularity, running it for billions of years on world after world, and almost never producing anything that looks at the sky and wonders about its neighbors, almost never producing anything like us. If that is the picture, if the K2-18b signal is confirmed, and if the silence continues, then intelligence is the rare thing. Not life. Intelligence.
And we are sitting inside the rare outcome, aware of the ocean 120 light years away, aware that it probably cannot be aware of us, aware that the rarity of that awareness makes it difficult to quantify and impossible to share with the things that would most change the conversation. The signal from K2-18b and the silence from everywhere else together describe a universe that is full of life and almost entirely alone in any meaningful sense of awareness. That is stranger and more interesting than either a lifeless universe or a universe buzzing with communication. It is the universe that the current data, taken together, most strongly suggests. Something that gets lost in the coverage of K2-18b is how much the discovery depends on decisions made 40 years ago by people who had no idea this planet existed.
The infrared detectors in the James Webb Space Telescope's near-infrared spectrograph, the instrument that captured the K2-18b transmission spectrum, descend from technology developed in the 1980s for military satellites and adapted for astronomical use in the 1990s. The specific detector arrays, mercury-cadmium telluride, were selected for Webb through a competitive process in the early 2000s. The engineers who built them were solving an engineering problem, not thinking about dimethyl sulfide in exoplanet atmospheres. There was no K2-18b in the catalogue. There were no confirmed exoplanets at all when the foundational detector technology was being developed. This is how scientific discovery actually works. The tools are built for reasons that seem adequate at the time, the capabilities accumulate over decades, and then at some unpredictable moment, a target appears that the tools were, inadvertently, perfectly suited to interrogate.
The James Webb Space Telescope was not designed to detect biosignatures on Hycean worlds. Hycean worlds were not a recognized category when the telescope was approved. The sensitivity required to measure dimethyl sulfide at K218b's distance was not on any specification sheet from 2003. And yet, here we are. The spectral resolution question: what Webb can and cannot separate. One of the technical details that matters for evaluating the K2-18b claim is the spectral resolution of the James Webb Space Telescope's instruments. Resolution in spectroscopy refers to how finely the telescope can distinguish between adjacent wavelengths, how close two spectral features can be before the instrument smears them together into a single, indistinguishable blob. The NIR-spec instrument, which covers wavelengths from roughly 1 to 5 micrometers, operates at resolutions up to about 2,700 in its highest resolution mode. That means it can distinguish features separated by about 1 part in 2,700 of the central wavelength. For a wavelength of 3 micrometers, that translates to a minimum separable feature width of about 1 nanometer. Dimethyl sulfide has absorption features in this range, but so do several other sulfur compounds and some organic molecules. At resolution 2,700, many molecular features can be separated, but not all. Some molecules have overlapping absorption features that cannot be distinguished even at Webb's best resolution. Identifying dimethyl sulfide specifically, rather than a family of related sulfur compounds, requires both high spectral resolution and high signal-to-noise, and relies on the specific pattern of multiple absorption features across a range of wavelengths, rather than any single feature. The team published their identification of dimethyl sulfide as a best fit molecule to the observed spectrum given a specific atmospheric model. Different atmospheric models, different assumed temperature profiles, different assumed mixing ratios for other constituents, produce different best fit molecules. This model dependence is one of the sources of uncertainty that independent groups are currently working through with the same data. The history of the habitable zone concept and why K2-18b complicates it.
The habitable zone, sometimes called the Goldilocks zone, is a concept that has shaped planetary science since the 1960s, when Stephen Hart and Michael Hart developed early quantitative versions of it. The idea is simple. There is a range of orbital distances from a star within which a planet could maintain liquid water on its surface, given reasonable assumptions about atmospheric composition and pressure. The concept has been refined many times since.
The boundaries of the habitable zone depend on the type of star, the mass of the planet, the composition of the atmosphere, and what assumptions you make about climate feedbacks. The current standard reference is the work of Ravi Kumar Kopparapu and colleagues, published in 2013, which extended the classical calculations to account for updated water vapor absorption data. K2-18b sits within Kopparapu's habitable zone boundaries for its star. But K2-18b also illustrates how the habitable zone concept can be misleading. The habitable zone was developed with Earth-like planets in mind: rocky surfaces, nitrogen-oxygen atmospheres, water on the surface. K2-18b, with its hydrogen atmosphere and predicted subsurface ocean, would not fit the assumptions that went into the habitable zone definition. Yet, it may be the most compelling habitable world candidate we have found. This suggests the habitable zone, while useful as a first filter, is not a sufficient criterion for habitability, or even for a meaningful search. Planets outside the classical habitable zone might host life in subsurface oceans, warmed by radioactive decay or tidal heating. Planets inside it might be too geologically dead to maintain the chemistry life requires. The habitable zone points you roughly in the right direction. It does not guarantee anything about what you find there. How the K2-18b discovery interacts with the ongoing SETI programs. The search for extraterrestrial intelligence has been running, in various forms, since Frank Drake pointed the Green Bank telescope at two nearby stars in 1960 and listened for radio signals in the first experiment, called Project Ozma. Nothing has been found in 65 years of searching, across progressively wider surveys with progressively more sensitive instruments. SETI and biosignature searches are often discussed as separate programs, and they are institutionally separate. Different funding streams, different research communities, different methodologies. But they are searching for the same thing from different angles. SETI looks for technological signatures, signals that could only be produced by intelligence. Biosignature searches look for chemical signatures that could be produced by any life, microbial or otherwise. The K2-18b result is irrelevant to SETI in one sense. Dimethyl sulfide is not a sign of intelligence, any more than Earth's dimethyl sulfide production is. But it is relevant to the broader search strategy in another sense. If microbial life is common, which a confirmed K2-18b detection would suggest, then the universe has been running biology experiments on billions of worlds for billions of years. The prior probability that some of those experiments produced intelligence becomes higher, not lower. More inhabited worlds means more chances for the evolutionary trajectory that leads to intelligence, even if that trajectory is rare. The SETI community has noticed the K2-18b signal. The Breakthrough Listen program, the most well-funded SETI effort in history, backed by Yuri Milner's breakthrough initiatives, has K2-18b on its candidate list for targeted radio and optical searches. Not because dimethyl sulfide implies radio transmitters, but because a world that might host any life is worth checking for every kind of signal we know how to detect, the specific star and what it contributes. K2-18 itself is worth understanding in some detail, because the star's properties directly determine what the planet experiences, and therefore what life on the planet would have to cope with. K2-18 is a red dwarf of spectral type M2.5. Its mass is about 33% of the Sun's mass. Its luminosity is about 2.5% of the Sun's. Its surface temperature is around 3400 Kelvin, compared to the Sun's roughly 5800 Kelvin. It is dimmer, cooler, and smaller in every dimension than our Sun, but also, crucially, far longer lived.
The Sun will exhaust its hydrogen fuel in roughly another 5 billion years. K2-18, burning so much more slowly, has a main sequence lifetime of roughly 50 to 100 billion years. It is barely middle-aged by stellar standards. That longevity matters for life. Life on Earth took several billion years to produce organisms capable of significantly altering the planet's atmosphere. If the timescale for life's development is similar elsewhere, then the long-lived red dwarf system gives life more time. K2-18b has had its current configuration for what is estimated to be several billion years.
If life started in that ocean early in the planet's history, it has had enormous time to diversify, to evolve metabolic pathways, to fill every available ecological niche. The star also matters for the photochemistry of the atmosphere. The spectrum of K2-18 is shifted far toward the red and infrared compared to the Sun. The ultraviolet flux is much lower. This affects what photochemical reactions can happen in the atmosphere. Some reactions that are driven by UV light on Earth would be suppressed around K2-18, which could allow molecules like dimethyl sulfide to accumulate to higher concentrations before being photo-destroyed. That is a point in favor of biological interpretation. The photochemical destruction rate may be slow enough that even modest biological production could maintain a detectable atmospheric signal. Or it could mean that some unusual abiotic photochemistry operating in that UV-poor environment can accumulate sulfur compounds that would be quickly destroyed on Earth.
The photochemistry is being modeled. The outcome of that modeling matters enormously. What the transit geometry tells us and what it hides. K2-18b transits its star from Earth's perspective. It passes between us and the star in a geometry that makes the transmission spectroscopy possible. The transit probability is not high for any given planet. It requires a specific alignment between the planet's orbit and the line of sight from Earth. That K2-18b transits is a piece of luck that makes it accessible to current methods. During a transit, the planet's disk blocks a fraction of the stellar light, about 1.4% in K2-18b's case, given its size relative to the star. The atmosphere adds a thin ring around the planet that is partially transparent to starlight, and that is what the spectrograph is reading. The signal is tiny. We are measuring the differential absorption of less than 1% of the stellar light by a layer of atmosphere a few hundred kilometers thick around a planet 120 light-years away. The transit lasts a few hours. During those hours, the James Webb Space Telescope must hold its pointing steady to the required precision, read out its detectors repeatedly, and accumulate enough photons to beat down the noise to the level where molecular features are detectable. Each transit observation produces one data point in the cumulative spectrum. Multiple transits, Madhusudan's team has used several, are needed to build the signal-to-noise required to identify trace molecules. What the transit geometry hides is most of the planet. We see the atmosphere at the terminator, the boundary between day and night on the tidally locked world, the narrow ring where the limb of the planet meets space. The dayside atmosphere, the nightside atmosphere, the ocean surface if there is one, the floor of the ocean. None of these are accessible. Transmission spectroscopy gives us a one-dimensional average through the thinnest part of the atmosphere at the terminator. It is a remarkable amount of information given the distances involved. It is also a very incomplete picture of what the planet actually is. The naming problem. What would we call them? If life is confirmed on K2-18b, a naming question will eventually arise. Not in any urgent practical sense. The organisms, if they exist, will not be aware of being named. But naming is a human act, with consequences for how we think about things. The category of life on Earth goes by many names at different levels of organization. Bacteria, archaea, eukaryotes, kingdoms, phyla, species. All of those names assume Earth biology. They assume DNA, cells, membranes, specific metabolic pathways. Life on K2-18b, if it exists, may or may not share any of those features. If it uses dimethyl sulfide as we know it, It has sulfur in its biochemistry. Beyond that, we know nothing. Whether it uses nucleic acids, whether it has cells, whether it reproduces through anything analogous to cell division, all unknown, and perhaps unknowable with current technology. The naming problem is a symptom of a deeper conceptual challenge. Our entire framework for categorizing life is built from one example. We have a taxonomy, a phylogenetics, a biochemistry, all of which describe variations within a single tree of life that sprouted from a single origin event on Earth. Everything alive today is related to everything else alive today. K2-18b life, if independently originated, is not on that tree. It is not a domain, not a kingdom, not a phylum. It is something for which we currently have no category, because we built all our categories from the one example we had. The discovery of a second example of life would require not just a news headline, but a fundamental reorganization of how biology thinks about itself. The time between now and knowing. The James Webb Space Telescope has a finite operational lifetime. It was designed for a 10-year mission, with fuel reserves that the precision of its launch may have extended to 20 years or more. The telescope launched in late 2021. If it operates until the early 2040s, there is roughly 15 to 20 years of remaining operational time. Within that window, K2-18b can be observed many more times. Each transit produces more data. The statistical significance of the dimethyl sulfide signal, currently at 2 to 3 sigma, will either increase or plateau or fade with additional observations. If it increases to 5 sigma over the next 5 years of observations, the signal is real. If it fades or stabilizes at low significance, the current detection was likely noise or an artifact. Parallel to this, the atmospheric chemistry modeling will mature. The laboratory spectroscopy of relevant molecules will be completed. The abiotic production question will be addressed more thoroughly. The independent analysis of the existing data by other groups will converge or diverge. The full picture will become clearer. Not through a single definitive moment, but through gradual accumulation of evidence pointing in one direction or the other. We are probably 5 to 10 years away from having enough to say something definitive. Not, "we have found life," in the press conference sense, but the balance of evidence strongly favors biological or abiotic origin. That is the realistic timeline. 5 to 10 years is short by the standards of the history of science. It is long by the standards of human impatience. But it is the actual window in which the answer to one of the oldest questions human beings have ever asked will either open or close on K2-18b. What it means to be alive at this particular moment. The search for extraterrestrial life has been running for as long as human beings have looked at the sky and wondered. Ancient philosophers speculated about it. Medieval theologians argued about it. Enlightenment scientists mapped other planets and imagined inhabitants. The 20th century gave the speculation instruments, radio telescopes, spacecraft, eventually exoplanet detection. And now, in the 2020s, the instruments have found something. Not proof, a candidate, but a candidate with more substance than any previous candidate has had. A specific planet, a specific molecule, a specific telescope, a specific team, specific data that has been published and checked and not explained away. The people alive right now are the first people in human history to occupy this particular epistemic position, past the point of pure speculation, past the point of zero evidence, in a period when a real signal exists in real data from a real planet, and the technology to test it further already exists and is operating. No previous generation had this. Future generations will have the answer, one way or the other. This generation has the signal and the uncertainty, and the work of testing it. That is an unusual place to be. The question has been asked for millennia. The answer is being measured right now, in numbers flowing from a telescope 1 million miles from Earth to servers where people are reading them. The interval between the asking and the answering has never been shorter. The instruments have never been better. The candidate has never been more serious. A few more things before the honest reckoning. The K2-18b story is not just about one planet and one molecule.
It is about what the act of searching does to the searcher. 65 years of SETI produced no confirmed signal, but produced an entire scientific community, an entire set of methodologies, an entire vocabulary for thinking about life beyond Earth. The James Webb Space Telescope was not built to find biosignatures. It was built to see the early universe, to study galaxy formation, to characterize the atmospheres of known exoplanets in general. The fact that it is now the primary instrument in the most important astrobiological search in history is a consequence of its capabilities meeting a moment it was not designed for. Science rarely goes where it plans to go. It goes where the instruments point and the data leads. The people who will ultimately confirm or refute the K2-18b biosignature may not yet be in graduate school. The instruments that confirm it may not yet exist. The theoretical framework that makes sense of it may not yet be written. What exists right now is the signal, the uncertainty, the work, and the waiting. And a generation of scientists who know they are the first to stand in this specific position, with this specific data, asking this specific question, with tools that can actually begin to answer it. The honest final reckoning. 3 categories. What is firmly established is the following. The James Webb Space Telescope has detected, in the transmission spectrum of K2-18b during multiple transit observations, spectral features consistent with carbon dioxide, methane, and dimethyl sulfide. The carbon dioxide and methane detections are considered robust by the community. The dimethyl sulfide detection stands at approximately 2 to 3 sigma statistical significance, real but below the conventional discovery threshold. K2-18b orbits within the habitable zone of its red dwarf host star, K2-18, located approximately 120 light-years away in the constellation Leo. The planet has a radius roughly 2.5 times Earth's and a mass approximately 8.5 times Earth's. Interior models consistent with these measurements allow for a Hessian configuration, a hydrogen-dominated atmosphere above a potential global liquid water ocean. On Earth, dimethyl sulfide is produced almost exclusively by biological processes, primarily marine microorganisms. The molecule has been detected at these concentrations in no other exoplanet atmosphere. The James Webb Space Telescope is the most sensitive instrument ever deployed for this kind of measurement. The team that published the results, led by Niku Madhusudan at Cambridge, is among the leading groups in exoplanet atmospheric characterization and has applied conservative interpretation standards throughout. What is actively debated is the following. Whether the dimethyl sulfide signal will strengthen to discovery-level significance with additional observations or reveal itself as a statistical artifact or instrumental systematic. Whether abiotic photochemical processes in a hydrogen-rich atmosphere irradiated by a red dwarf can produce dimethyl sulfide at concentrations sufficient to explain the observed signal. Current models disagree, with some groups finding plausible abiotic pathways and others finding them insufficient by orders of magnitude. Whether the K2-18b interior is actually configured as a Hessian world, or whether alternative interior models without a liquid water ocean are equally consistent with the mass radius data. Whether the spectral features attributed to dimethyl sulfide might partially overlap with features from other, less well-characterized sulfur or organic compounds whose spectroscopic properties at Hessian world pressures and temperatures are not fully measured. How much weight to give the biosignature candidate relative to the prior probability of finding life at the first seriously habitable zone planet characterized at this level of detail, which could reflect either genuine discovery or observational selection bias, or both. Whether K2-18b's red dwarf host star's flaring history and UV environment
are compatible with maintaining a habitable ocean over geological timescales. The correct allocation of future James Webb Space Telescope time between more observations of K2-18b and characterization of other Hycean world candidates. What cannot yet be known is the following: whether life exists on K2-18b, whether life exists anywhere beyond Earth, whether the origin of life is a common or rare event given suitable conditions, which is the question that a confirmed K2-18b biosignature would most sharply illuminate.
Whether the biochemistry of any potential K2-18b life uses molecules, structures, or processes analogous to Earth biology, or something entirely different. Whether the Hycean ocean, if it exists, has the energy sources, hydrothermal, photosynthetic, or other, necessary to sustain a biosphere capable of maintaining a detectable atmospheric signature. Whether the great filter that resolves the Fermi paradox lies behind us or ahead of us, a question whose answer depends on how common microbial life turns out to be, which the K2-18b data is beginning to address but is not yet resolved. What confirmation of extraterrestrial life would do to human societies, institutions, belief systems, and self-understanding at a civilizational scale? Whether future telescopes, the extremely large telescope, the Habitable Worlds Observatory, technologies not yet designed, will confirm the K2-18b signal, refute it, or find something more unambiguous elsewhere.
And what the universe looked like on K2-18b for the billions of years before any species on Earth learned to build instruments sensitive enough to read its atmosphere. The dimethyl sulfide is in the data. The data is real. The question it raises is the oldest question. The answer is not here yet, but the measurement that will produce it is running.